CDKL5 is a rare disorder with about 1,200 documented cases. It is caused by mutations within the CDKL5 gene that cause a deficiency in the protein. The CDKL5 gene is found on the X chromosome, which is why we find that more girls affected than boys. It causes seizures within the first 6 months to a year of life, which results in severe developmental delays as well as motor function delays. The Loulou Foundation was founded in 2015 with the goal of accelerating research in biological understanding of CDKL5 deficiency, as well as therapeutic approaches to the disease. The Orphan Disease Center is a relatively new program established at the University of Pennsylvania with the goal of progressing translational research in rare disease and also bringing communities together around rare disease. And that community exists as part of the intersection between patient advocacy groups, academics, biotech, and pharma as well. Through the partnership with the Loulou Foundation and the Orphan Disease Center at UPenn, the groups are trying to build bridges across academia, biotech, pharma, and patient advocacy by accelerating research, and that can be done by identifying gaps and obstacles within the clinical translational path that have traditionally existed for rare disease, as well as accelerating timelines for academic research and incentivizing groups to be interested within the CDKL5 deficiency space. So through this partnership, we've recently funded 11 new grants within the CDKL5 deficiency field. So we're hoping to really accelerate translational research. Our current grantees have a range of different backgrounds and projects they're focusing on, from drug repurposing to development of better animal models to understand the disease. We're also interacting with biopharm to try to understand their interest in rare disease. And it's an exciting time because biopharm is starting to recognize rare disease as the unmet need it is. And so we're seeing that interest rise across a number of disorders, including CDKL5. So we're identifying partners in academia to accelerate translational research and also identifying partners and building bridges between the patient advocacy groups and biopharm interests.
At the University of Edinburgh, we have an incredibly rich and vibrant neuroscience community that includes the Patrick Wild Center for Research into Autism, Intellectual Disability, and Fragile X Syndrome, as well as the Muir Maxwell Center for Research into Epilepsies. Recently, as a result of a 3-year grant from the Loulou Foundation, we've started to really try to understand the fundamental basis of CDKL5. This is a collaborative research project between my lab and the labs of Professor David Wiley, Giles Hardingham, and Michael Cousin, as well as our collaborators John Rouse at the University of Dundee. The fundamental goal of this research is really twofold. First, we want to understand the basic mechanisms of brain development that are regulated by CDKL5 and crucially understand what goes wrong when CDKL5 is missing. By understanding the fundamental basis of CDKL5, we can hope to develop novel theories that will eventually lead to clinical interventions. To achieve this, we really have to understand the fundamental role of CDKL5 across levels, from the synapse to the cell to the circuit, and ultimately how it controls brain function and behavior. By its very nature, this type of research takes a long time. It will be quite a while before we actually get to the point of developing novel therapeutics that will make a real difference for the families with CDKL5. Therefore, as a second strand of this research, we're really trying to understand how CDKL5 relates to other neurodevelopmental disorders so that we can piggyback off the knowledge that's already been gained from these disorders, as well as the clinical therapies that are already in trial for these disorders. By doing this, we hope to be able to understand CDKL5 in a way that we can take these therapeutic interventions and bring them to the clinic much more quickly.
CDKL5 disorder is a special orphan disease with a known genetic cause, meaning we know CDKL5 is the gene responsible for the syndrome. Fortunately, the mouse and human versions of the CDKL5 gene are very similar to each other, especially at the protein level. This has allowed us to make a mouse carrying the exact same CDKL5 mutation as human patients, and thus modeling the disorder in mice. Partially sponsored by the International Foundation for CDKL5 Research, IFCR, we have generated the first mouse model of CDKL5 carrying a patient mutation. These mice show behavioral phenotypes mimicking multiple symptoms of a patient, thus setting up the stage for us to investigate CDKL5 disorder using mouse genetics. The 2-year grant from the Loulou Foundation, together with the International Foundation for CDKL5 Research, has truly made our research objective possible. In these grants, we aim to solve 3 fundamental questions. We want to know where in the brain CDKL5 is essential. This is important because the brain has many different types of cells, and CDKL5 may be more important in some parts of the brain than others. We also want to know when CDKL5 is required for brain development. This will tell us about the biological processes that underlie the onset of the disorder. Finally, we will attempt to reverse CDKL5 disorder-related phenotypes in mice by restoring CDKL5 function. This arm of the project will help us determine both the time window of therapy and the level of CDKL5 needed to reverse the phenotypes. I feel very fortunate to have the Loulou Foundation sponsoring our research, and we're very excited to have the opportunity to work with the rest of the CDKL5 community and the CDKL5 families. Our goal is to first establish a working mouse model of the disorder, determine the measurable outcomes, and ultimately setting up the platform to test the potential therapies for CDKL5 disorder in animals.
My team and myself are very keen to get to the bottom of why mutations in the CDKL5 gene cause the symptoms associated with CDKL5 deficiency. I think that to develop effective treatments for children with CDKL5 deficiency, we need to understand what it is that CDKL5 does normally inside cells in our bodies to keep them healthy. Knowing the normal functions of CDKL5 would help us to figure out what goes wrong in CDKL5 deficiency. So how do we find out what CDKL5 does normally inside our cells to allow our bodies to function normally? Well, we know that CDKL5 is what we call a kinase. So kinases are tiny machines inside our cells, and what they do is to modify other proteins in a way that's important to keep cells healthy. There are lots of kinases in our cells, and we know for a lot of the kinases which proteins they modify inside cells. The problem is that for CDKL5, we do not know which proteins it modifies in cells. And finding the proteins that CDKL5 modifies is the key to understanding what it does normally. We think that the symptoms associated with CDKL5 deficiency arise from a failure to modify key proteins inside cells. So very simply, our mission is to find the proteins that CDKL5 modifies inside our cells. My laboratory is based at the MRC protein phosphorylation unit at the University of Dundee, and our unit has a long history of studying kinases and a long history of identifying the proteins modified by individual protein kinases. We're interested in kinases mutated in disease, and just recently teams here have been working on a protein mutated in Parkinson's disease called PINK1. PINK1 is a kinase. And 2 years ago, teams here identified the first substrates of the PINK1 kinases that has elucidated how mutations in this kinase cause Parkinson's disease. So what we want to do now is to use the same methodologies for PINK1 for CDKL5. We want to apply state-of-the-art technologies, including a technique called mass spectrometry, to identify the proteins inside cells that are modified by CDKL5. So we hope in this way to find out what CDKL5 does under normal circumstances, so we can find out what goes wrong in CDKL5 deficiency. Because it's possible that its failure to modify key specific proteins inside cells that don't have CDKL5 that are responsible for the symptoms that we see in CDKL5 deficiency. The experiments that we're doing to identify the CDKL5 modified proteins is funded by the Loulou Foundation, and we're very grateful to Loulou for the funding for these experiments. We're going to be collaborating with Loulou-funded investigators at the University of Edinburgh, and these include Professor Peter Kind and Professor Giles Hardingham.